Good day, everyone. I am Mr. John Angelo B. Almazar, and I will be reporting protein metabolism. So, at the end of this learning session, the students or the listeners may able to describe what protein is in terms of functions, structure, and types. Compare and contrast protein anabolism and catabolism. Identify the processes involved in protein anabolism and catabolism, and describe how the body digests and metabolizes proteins. There are different types of proteins that can be found in our body. Like for example, the structural protein that can be found on the hair and the nails. Contractile protein that can be found on the muscles. Transport proteins that can be found on the blood, which transport ions and other nutrients in our body. The hormones, the ion channels which are responsible for the entrance and exits of some materials inside and outside the cell membrane. Antibodies and cell receptors which are responsible for immune response, and enzymes which are responsible for catalytic and metabolic activities in the cellular level. Proteins can also be found on different parts of the cells, like for example in the mitochondrion. So the membrane of the mitochondrion possess proteins. The ribosome is also made up of protein and RNA. The plasma membrane consists of protein channels which are responsible for the entrance and exits of some materials. Inside the nucleus, we can also find the DNA, which is genetic blueprint of life, which are wrapped around the histone, and histone is an example of a protein or is a protein itself. The microtubules are also made up of proteins and other parts of the cells. We have here an example of structural formula of protein. Remember that proteins are composed of amino acids joined by the peptide bonds. So we have here three amino acids joined by peptide bonds, and each of the amino acids are composed of the amino group, the carboxyl group, the central carbon, and very unique side chains, so that made it very different from each other. So amino acids and the other amino acid will be different from each other because of very unique side chains. So for example, here we have phenylalanine, the glutamine, and cysteine. Though they have the same amino group, carboxyl group, central carbon, but very unique in terms of the side chains. So that made it very unique from other amino acids. So the amino acids are joined together with the peptide bond, forming very long polypeptide chains, and that is what you call the primary structure. But then when it bends or folded, it becomes the secondary structure. And then further folding of the secondary structure will lead to the tertiary structure, and further folding of that tertiary structure will become quaternary structure, which made up of the conformation or the shape of the protein. So the shape of the protein will give the specific function of, or will dictate the specific function of that protein. So like for example, here we have the ligand and the binding site from the protein. So the folded protein have very specific and very unique binding site. Therefore, it can only recognize a ligand which is very specific and very fit to that kind of binding site of the folded protein. There is many different types of amino acids, and some of them are being manufactured or being synthesized naturally in the body or in the cell. However, there are some amino acids which are we called essential amino acids that cannot be synthesized naturally in the body. Instead, we need that to include in our diet. So like for example, the histidine, we need to take soy protein, eggs, parmesan, sesame, and peanuts in order for us to have that kind of protein. The isoleucine, the leucine, lysine, methionine, cysteine, phenylalanine, tyrosine, threonine, tryptophan, and valine. So those are what we call essential amino acids, and we need that to incorporate in our diet so that we can have those types of amino acids in our body. So another term that we have to discuss or we have to know is the metabolism. So when we say metabolism, so that is the term that is used to describe all chemical reactions involved in maintaining the life state of the cell and the organism. So the energy of the organism depends upon on metabolic processes. So like for example here on a meme example, so it might look like I am doing nothing, but at the cellular level, I am really quite busy because there are so many metabolic activities that is happening inside each cells of that dog sleeping. So therefore, though it looks like not busy, but in cellular level, it is very busy. So we have two kinds of metabolism, the catabolism and the anabolism. When we say catabolism, that is the breaking down of molecules to obtain energy. And when we say anabolism, we synthesize of all compounds needed by the cells. So an anabolic pathway or anabolic metabolic pathways, small molecules are assembled into large one. Like for example, monosaccharide, and then we bind together another monosaccharide. We have now the disaccharide, and another monosaccharide. Then we have now the carbohydrate. In our case, we have amino acid plus peptide bond, then another amino acid, then another peptide bond. So we have now proteins. But when you say catabolic activity or catabolic metabolic pathways, so that is different from the anabolism. So large molecules are break down into small one, and then the energy is being released. So protein metabolism denotes the various biochemical processes responsible for the synthesis of proteins and amino acids, so that is on the form of anabolism, and the breakdown of proteins by catabolism. So there are different steps when we say protein metabolism, but the protein metabolisms start on our stomach, on the digestive system, wherein we have different enzymes responsible for the digestion of proteins, like for example, the proteases, the pepsin. We have also the trypsin, chymotrypsin, elastase, carboxypeptidase. So those are examples of the enzyme that is being used in order to break down protein or to denature proteins. So after on the digestive tract of the body, so it will be absorbed in our circulatory system, and it will be passed on the liver. Then the liver will pass it again to the specific parts of the body through our circulatory system so that it can be used by the body. So the protein metabolism involves different types of processes, like for example, the transcription, the translation, the post-transcriptional modification, and the folding of proteins so that it can be formed into a very specific conformation of the protein so that it can perform very specific function of it. So let us recall the central dogma of the molecular biology. So remember, the DNA is replicated to form another DNA. And then when the DNA is already replicated, it might be transcribed to form the RNA. So, or the ribonucleic acid. And that RNA, which is a transcript, so it can be translated to form protein. And that is the central dogma of the molecular biology. DNA, then DNA to RNA, then the RNA will be translated to form the protein. The first part of the synthesis of the protein is the transcription. So here in transcription, the RNA polymerase reads a DNA strand and produces an mRNA, which is a messenger ribonucleic acid strand that can be further translated. In order to initiate transcription, the DNA segment that is to be transcribed must be accessible. So remember that the DNA is tightly packed because of the presence of two hydrogen bonds on adenine and thymine, and three hydrogen bonds on the guanine and cytosine. So therefore, it should be unzipped first so that there will be an accession to the DNA strand. Once a DNA segment is accessible, the RNA polymerase can begin to transcribe the coding DNA strand by pairing RNA nucleotides to the template DNA strand. During the initial transcription phase, the RNA polymerase searches for the promoter region. Ano? So this promoter region on the DNA template strand, once the RNA polymerase binds to this region, it begins to read the template of the DNA strand in 3' to 5' direction. The RNA polymerase attaches RNA bases complementary to the template DNA strand, but instead of the thymine, uracil will be used instead. The new nucleotide bases are bonded to each other covalently. The new bases eventually dissociate from the DNA bases but stay linked to each other, forming a new mRNA strand. This mRNA strand is synthesized in the 5' to 3' direction. Once the RNA reaches a terminator sequence, it disassociates from the DNA template strand and terminates the mRNA sequence as well. Transcription is regulated by transcription factors. These transcription factors are proteins that binds to regulatory sequences of the DNA strand, such as promoter regions or operator regions. The protein bound to these regions can either directly halt or allow the RNA polymerase to read the DNA strand, or can signal other proteins to halt or allow the RNA polymerase reading. Next process is what we call the translation, wherein the messenger ribonucleic acid or the mRNA is being translated into specific amino acid and further elongated to form proteins. So during this stage, ribosomes convert a sequence of mRNA to amino acid sequence. Remember that the ribosome is the one that is responsible for translation of proteins. So each 3-base pair long segment of mRNA is what you call codon, which corresponds to one amino acid or stop signal. Amino acids can have multiple codons that corresponds to them. Ribosomes do not directly attach amino acids to mRNA codon. They must utilize the transfer ribonucleic acids or the tRNA as well. This tRNA can bind to amino acid and contain an anticodon which can hydrogen bind to an mRNA codon. This process of binding an amino acid to tRNA is known as the tRNA charging. So here, the enzyme amino acetyl tRNA synthetase catalyzes two reactions. The first one, it attaches to the AMP molecule, and the second reaction cleaves to amino acid AMP, producing the energy to join the amino acids to the tRNA molecule. So the ribosome enters to the elongation process, which follows a repetitive cycle. So the ribosomes have two subunits, the one large, and the other one is the small, ano? And these larger subunits have the A amino acid, the P peptidyl, and E exit. Okay, as you can see on the video, so it start to a specific site, going to another site, and then going to another site, so that is being repeated again and again until the protein is elongated. And the formation of peptide bond requires as well as an input of energy. So remember that amino acid can attach to another amino acid by having the peptide bond. So, and amino acid, peptide bond, amino acid, then eventually we have protein. So next part is the post-translational modification. So once the peptide bond chain is synthesized, it still must be modified. So we have two parts of the post-translational modification. So the first one, we have the methylation. So the methylation reduces the ability of some amino acids to form hydrogen bonds so that arginine and lysine that are methylated have different properties than other standard counterparts. So, and another one, we have also the phosphorylation, which are often occurs to serine, threonine, and tyrosine, and involves replacing a hydrogen on a alcohol group at the terminus of the R group with the phosphate group. So, another part of the synthesis of the protein, which is also very important, is the protein folding. So here, the polypeptide chain in the cell does not have to stay linear. So it cannot stay linear because that is very long. It can become branched or folded in one itself. Polypeptide chains fold in a particular manner depending on the solutions they are in. The fact that all amino acids contain R groups with different properties is the main reason proteins fold. So therefore, that is the main reason, the containment of the R groups. So that R group actually is a hydrophilic in environments such as cytosol, and the hydrophobic amino acids will concentrate at the core of the protein, while the hydrophilic amino acids will be on the exterior. So these entropically favorable since the water molecules can move much more freely around the hydrophilic amino acids than the hydrophobic amino acids. So in hydrophobic amino acids, the hydrophilic amino acids will concentrate to the core of the protein. Then while the hydrophobic amino acids will be on exterior, since the new interactions between hydrophilic amino acids are stronger than the hydrophilic interaction. So this is what you call the entaphilically favorable. So here we have the primary structure, a very long chain of amino acids, and then it is being folded to form the secondary, and then another fold to form the tertiary structure, and the final product, that is the quaternary structure, which made up the shape of the protein or the conformation of the protein, and that made the function of the protein. So that dictates the functions of the protein. So after discussion of the synthesis of the protein, we also have the protein breaking down, the breaking down of the protein, and that is what you call the protein catabolism. So it is the process by which proteins are broken down to their amino acids. So this is called proteolysis and can be followed by further amino acids degradation. Protein catabolism via enzymes. So catabolism may take place through the use of enzymes, like for example here the proteases. So proteases can be found on animal digestive tract and human digestive tract. So these proteases breaks down protein into amino acids, but originally thought to only disrupt enzymatic reaction, proteases, also known as the peptidases, actually help by catabolizing proteins through cleavage and creating new proteins that were not present before. So aside from disrupting it, so it is also being used to create new proteins. Proteases also helps to regulate the metabolic pathways. One way they do this is to cleave enzymes in pathways that do not need to be running. This helps to conserve as much energy as possible and to avoid futile cycles. So the futile cycles occurs when the catabolic and abolic pathways are both in effect at the same time and the rate of for the same reactions. So since the intermediates being created are consumed, the body makes no net gains, energy is lost through the butyl cycles, and the proteases prevent these cycles from occurring by altering the rate of one of the pathways or by cleaving a key enzyme that cleaving the enzyme that they can stop of the pathways, that stop one of the pathways. Proteases are also non-specific when binding to substrate, allowing for great amounts of diversity inside the cells and other proteins as they can cleave much easier in an energy efficient manner. So, for example, here, so this is the possible mechanism for aspartyl protease cleaving a peptide bond. So because many proteases are non-specific, they are highly regulated in the cells. Without regulation, proteases will destroy many essential proteins for physiological processes. So one way the body regulates proteases is through proteases inhibitors. Protease inhibitors can be other proteins, small peptides, or molecules. So those are there are types of the protease inhibitors, the reversible and the irreversible proteases. Aside from proteases, we have also enzymes, exopeptidases, and endopeptidases. But the proteases can be released on the stomach of the animal and human. However, these exopeptidases can be synthesized on the small intestines or can be released on the small intestines. So these are enzymes that can cleave at the end of amino acid side chains mostly through the addition of water. So these enzymes have two classes, the aminopeptidases and carboxypeptidases. So aminopeptidases are enzymes that remove amino acids from the amino terminus of the protein. So they are present in all life form and they are all crucial for survival since they do many cellular tasks in order to maintain the stability. This form of peptidases are zinc metalloenzymes, and it is inhibited by the transition state analog. So this analog is similar to the actual transition state. So it can make the enzyme bind to it instead an actual transition state, thus preventing substrate binding and decreasing reaction rates. So the carboxypeptidase cleave at the carboxyl end of the protein. While they can catabolize as protein, they are most often used as post-transcriptional modification. So another enzyme here, we have the endopeptidases. So these endopeptidases are enzymes that add water to an internal peptide bond in a peptide chain and break that bond. Three common endopeptidases that come from the pancreas are pepsin, trypsin, and chymotrypsin. So those enzymes are actually being manufactured by the pancreas for the metabolism of the protein. The chymotrypsin performs hydrolysis reaction that cleaves after aromatic residues. The main amino acids involved are serine, histidine, and aspartic acid. They all play a role in cleaving the peptide bond. These three amino acids are known as the catalytic triad, which means that these three must all be present in order to properly function. So those three should be present, the serine, histidine, and aspartic acid, so that there will be a catalytic activity, and that is what you call the catalytic triad. So those enzymes, proteases, exopeptidase, and endopeptidases are the enzymes that are responsible for breaking down of proteins. So again, the proteases are being released and manufactured on the stomach, while the exopeptidases and endopeptidases can be released on the small intestines and the pancreas, respectively. Aside from enzymes, protein can be catabolized by environmental changes. So like for example, the pH, the cellular proteins are held in relatively constant pH in order to prevent changes in the protonation state of the amino acid. Of course, sudden drop of the pH or increase of the pH can disrupt the protein. Some amino acids in the polypeptide chains can become protonated if the pKa of their R group is higher than the new pH. So one of the environmental changes that can disrupt protein or that can catabolize protein is pH. Another one is the temperature, like when you are cooking the egg. When you cook the egg white, you cannot turn it back to its original raw stage. Why? Because the proteins are already denatured. The protein albumin is already denatured. So as the temperature in the environment increases, molecules move faster. Hydrogen bonds and hydrophobic interactions are important in stabilizing proteins, in stabilizing forces in proteins. If the temperature rises, and the molecules containing these interactions are moving too fast, the interactions become compromised or even break. At high temperatures, these interactions cannot form, and as functional protein is denatured. When we say denaturation, that is breaking down of the conformation of the proteins. Let us now discuss digestion and metabolism of protein in vertebrates. The digestion and metabolism of protein start on the stomach, unlike the carbohydrates, which can start already on the mouth because of the presence of the salivary enzyme or the salivary amylase on the thiolin. But then the protein can only start breaking down on the stomach because when the protein rich foods enter the stomach, they are greeted by the mixture of the enzyme pepsin and hydrochloric acid. The latter produces an environment pH of 1.5 to 3.5 that denatures proteins within the food. And then this pepsin, which is an enzyme, cuts protein into smaller polypeptide chain and their constituents amino acid. When the food gastric juice mixture, which is what we call the chyme, enter the small intestine, the pancreas release sodium bicarbonate to neutralize the hydrochloric acid. This helps to protect the lining of the intestine from ulceration. The small intestine also release digestive hormones, including secretin, the CCK, which stimulates digestive processes to break down proteins further. So this secretin is also stimulates the pancreas to release sodium bicarbonate, and the pancreas release most of the digestive enzymes, including proteases like trypsin, chymotrypsin, and elastase, so which aid protein digestions. Together, all of these enzymes break complex proteins into smaller individual amino acids, which are then transported across the intestinal mucosa to be used to create new proteins or to be converted into fats or acetyl coenzyme A and used in the Krebs cycle. So remember that in the Krebs cycle, so there is already a formation of energy. In order to avoid breaking down of proteins that make up the pancreas and small intestine, pancreatic enzymes are released as inactive proenzymes, so that are only inactivated in the small intestines. So these pancreas vesicles store trypsin and chymotrypsin as trypsinogen and chymotrypsinogen. Once released into the small intestine, an enzyme found in the wall of the small intestines called enterokinase binds to trypsinogen and converts it into active form. So this trypsin then binds to chymotrypsinogen uh, to convert into active chymotrypsin. No? So this chymotrypsin, okay, breaks down large proteins into smaller peptides, a process that is what you call the proteolysis. So when the protein is break down into smaller peptides, so that is what you call proteolysis. These smaller peptides are catabolized into their constituent amino acids, which are transported across the apical surface of the intestinal mucosa in the process that is mediated by sodium amino acid transport. So this transporter binds sodium and then bind the amino acid to transport it across the membrane. Another process of protein metabolism is what we call the urea cycle. So it is a set of biochemical reactions that produces urea from ammonium ions in order to prevent a toxic level of ammonium in the body. So it occurs primarily in the liver and to the lesser extent in the kidney. So remember then when we hear the word ammonium and urea, so we can associate it with urine. Okay, and in order to prevent from having a toxic level of that ammonium and urea, so it should be eliminated in our body. Prior to the urea cycle, ammonium ions are produced from breaking down of amino acids. In this reaction, an amine group or ammonium ion from the amino acid exchange with the keto group on another molecule. So this is what we call the transamination. Ano? In the urea cycle, ammonium is combined with carbon dioxide, resulting in urea and water. The urea is eliminated through the kidneys in the urine, and it can be excreted through urination. So in this reaction, an amine group of or ammonium ion from the amino acid is exchanged with keto group on another molecule. So this transamination event creates a molecule that is necessary for the Krebs cycle and an ammonium ion that enters into the urea cycle to be eliminated. To summarize, the general pathways of amino acid metabolism. So the proteins from the food may digested into amino acid and it can undergo two processes, anabolic ways and catabolic ways. On anabolic ways, it can synthesize the cell and the extracellular proteins, and it can also synthesize the peptide physiologically active substances to form proteins and peptides of the organisms. On the other hand, amino acids may undergo catabolic ways like transaminations, deaminations, and decarboxylation processes in which it can join with the it can produce urea and join with the carbon dioxide and water, and is being released to the process called urination, and that is in a form of urine. And some metabolites and glycolysis and Krebs cycles may also produce amino acids. Thank you everyone for listening, and if you have questions, so please feel free to ask on our group chat. Thank you.